Hey guys, hi, welcome to another problem from IBO previous year paper 2013, question number 8. Okay, here's an interesting question. Some substances need to be transported actively or passively from their site of synthesis, that is where they are made, to the location where they are active, where they are going to be used. The question is very simple. Indicate for which of the following substances if they are transported from the cytoplasm to the nucleus. Very important, from the cytoplasm to the nucleus. Okay, let's examine them one by one. Okay, also guys, you should be aware that it's not necessary that only one of the answers will be correct. Okay, so tRNAs, what are tRNAs? If you remember, tRNAs, they carry amino acids, right? So if we remember the central dogma says DNA is read into RNA and RNA is read into amino acid chain or a polypeptide. Now, the reading of RNA into amino acid chain, that is translation. Translation is accomplished by ribosomes, and ribosomes can only do it because there are specific tRNAs which carry specific amino acids, right? And what is the site of translation? Because we are going to talk about translation and nucleus, so that means we are talking about eukaryote, right? So that definitely means that here the translation is going to happen in cytoplasm, right? So they don't need to be transported from cytoplasm to the nucleus. So definitely not us. Histone proteins. Now what are histone proteins? If you remember, these are sort of basic proteins which help in the condensation of DNA, right? They help in condensation of DNA. That means they have to be inside the nucleus. That means that since in the eukaryote all the proteins are going to be synthesized in the cytoplasm, they need to be transported from the cytoplasm to the nucleus. So this is definitely our guy. Okay. Now nucleotides. What are nucleotides? So nucleotides are monomers. They are building blocks of DNA. What is a nucleotide? It's made up of a nitrogenous base, which could be A, T, G, or C. There goes your sugar. In case of DNA, it's deoxyribose, and there goes your phosphate. So this combination together is a nucleotide. Many nucleotides together into N. There goes your DNA. That means nucleotides need to go inside the nucleus where the DNA replication happens. What is DNA replication? Copying the DNA, right? So you need nucleotides to make a new strand. So that means this is also our guy because they need to be transported from the cytoplasm to the nucleus. ATP synthase subunits. Now, where do you find ATP synthase? If you remember, ATP synthase uses the H plus ion gradient. What's a gradient? Difference in the concentration of H plus ions. Now, this gradient can be used in two places. If you're talking about chloroplast, that means inside the lumen of thylakoid, we build a gradient. We deposit H plus ions, and these H plus ions drive the ATP pump. Okay, and also we deposit H plus ion in the intermembrane space. That is the space between the two membranes, outer and inner membrane of the mitochondria, right? And then once again, this H plus ion gradient is used to drive the ATP pumps in the inner membrane and make ATP. So both of these cases where we have the ATP synthase, the ATP pump, that's in the chloroplast or the mitochondria. Hence, we are not going to need it, right? That's not our guy. So in this case, you have two correct answers. That's B and C. Okay. So I hope you were able to learn something. And if you did, please do share these questions with your friends and your folks. Also, there's the book NCERT Map, which is going to help you visualize the concepts really easily. So go order a copy from Amazon. Take care. See you next time.